Hello everyone. Today now we are going to continue with the next class of anti-cancer drugs that are topoisomerase poisons. So before starting with this class of a drug, we should know what are topoisomerases. Topoisomerases are the enzymes that controls the degree of supercoiling in DNA and maintain the structure during the DNA replication and transcription to RNA. Now what do we mean by DNA supercoiling? Consider this is one strand of a DNA and this is the another strand of a DNA. I'll show you with the help of an example. This is, consider these are two different strands of DNA. This is purple color one is one strand and this one is another strand. Now here the coiling is taking place. So when the DNA supercoils, there is a tension produced. On this part of the DNA. You can see there is a tension produced on this part of the DNA, and due to which the supercoiling is taking place. This is the supercoiling. This is called as the supercoiling of DNA. Topoisomerases are of two types. Type 1 topoisomerase type 1 and topoisomerase type 2. Topoisomerase type 1 enzyme, it cleaves single strand of DNA. And resells it. Topoisomerase type 2 enzymes, they cleaves both the strands of DNA. Now we will just see how this takes place. Consider this is a supercoiling of the coiling of DNA. This way, and if the one strand of a DNA is cut, this part opens up and reseals here, this side. So the base pairs of this strand and this strand are now open for replication. This strand and this strand are now open for replication, and DNA polymerase helps to build up a new leading strand for both the DNA and replication take place. In case of topoisomerase 2 enzyme, both the strands in this type of coiling, both the strands of the DNA are cut, and this way the strands, the base pairs are exposed. Now, what are topoisomerase poisons or the anti-neoplastic drugs that functions as top poisons stimulate the DNA cleavage but inhibit the DNA resealing activity of the enzymes, leaving the DNA irreversibly damaged and unable to replicate. They are classified as first one, Camptothecins. Examples of Camptothecins are irinotecan, and topotecan. Second one, epidophylotoxins. Example is etopside. Then the last one is anthracycline, and the examples are doxorubicin and daunorubicin. Now we will see the mechanism of action of camptothecin. In this video we will be studying in detail the camptothecins, that is irinotecan and topotecan. Camptothecins inhibit topoisomerase 1 inhibitor in enzyme, and these two are the receptors for topoisomerase 2 enzyme. Next we will see the mechanism of action of camptothecins. How does this camptothecin work or inhibit topoisomerase 1 type of enzymes? So consider that this is one strand of DNA and this is another strand of DNA. and when the DNA is bound to the enzyme topoisomerase, this is the enzyme which has been bound to one strand of the DNA, it cleaves it. Okay, so this is the open part of the DNA which has been resealed. So this is the topoisomerase enzyme which has cleaved one strand of the DNA, and the resealing has taken place in this part. But how does this camptothecins work? Camptothecins work by if this is the strand of DNA and this one is the other strand, topoisomerase has cleaved the one strand of DNA this side. So this is an enzyme substrate complex. So what does these drugs do? The camptothecin, consider here I am writing the camptothecin as an inhibitor. So this inhibitor, it goes and binds to the enzyme substrate complex, and this further forms a ternary complex. Ternary complex. What is a ternary complex? The ternary complex is when the enzyme binds to the substrate and inhibitor at the same time. So this is an E for topoisomerase enzyme, substrate for DNA, and inhibitor which is our camptothecin. So this is a ternary complex formed here. Once this complex is formed, the DNA resealing is inhibited, and since the DNA resealing is inhibited, the DNA is not able to replicate and the cell may die. Now we'll see the structure and chemistry of camptothecins. Example of the drug is irinotecan. The structure is, it consists of a quinoline nucleus. So there is a quinoline nucleus which is fused with tetrahydropyrrole. This is a tetrahydropyrrole which is fused with the pyridine. This is a pyridine, and it further is fused with pyran ring. So there is a one more double bond O. Here it is OH. This is C2H5. At this position we have a C2H5 group, and in this position we have a piperidine moiety, which is there is one more piperidine group, which makes this drug highly basic. Then the structure of topotecan is it also has this quinoline nucleus. Then which is fused with a tetrahydropyrrole, which is further fused with a pyridine ring, and which is further fused with the pyrene ring. There is OH, C2H5 here, and the OH group here. The ninth position, it has a methyl group, which is further substituted with dimethylamine. We will number this. This is 1, this is 2, this is 3, 4, 5, 6, 7, it is 8, 9th and 10th. Similarly here also, the 9th position of topotecan and the 10th position of irinotecan consist of this basic groups. Which makes them water soluble. Salts. Irinotecan uses they are given in combination with fluorouracil as a first line therapy for the metastatic colorectal cancer. This is the first use. It is also given in combination with cisplatin in small cell and non-small cell lung cancer. The various side effects of this therapy are, I'll write down the side effects here. Irinotecan, side effects. The main side effect is myelosuppression, leukoneutropenia, then diarrhea, which can be fatal many times, then flushing, sweating, blurred vision, lacrimation, and sometimes bradycardia. Uses of topotecan are it is given IV for ovarian and small lung cancer, as a first line therapy. And the side effects are of topotecan are myelosuppression, neutropenia, thrombocytopenia, and anemia. So that was about the topoisomerase poisons, its type, classification, and a brief idea of camptothecins. Thank you for watching.